Hello, everyone, and welcome to today's webinar, Iterative Genome Engineering for Rapid Improvement of Protein Activity and Production. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Inscripta. To learn more, visit inscripta.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Eric Abbate, Director, Analytical Biochemistry, Applications Development, Inscripta. Eric, you may now begin your presentation.
Thank you for that introduction. So today I'm going to give you an introduction to Inscripta's Onyx platform and tell you about the iterative genome engineering workflow on that platform. Then I'm going to tell you about a few applications where we use this iterative genome editing in order to improve the function of a protein as well as the expression of a heterologously expressed protein in yeast. So there are a lot of challenges in the synthetic biology and the biological engineering space. So looking at something like forward engineering, where you're trying to produce more of a product or produce it faster, like heterologous protein production, it really requires high dimensional optimization. And unfortunately, we don't really know the rules of biology that allow us to go in and tinker with a genome in order to optimize the expression of a protein or a small molecule. In the reverse direction, when you're doing genome discovery, you're really trying to understand how the system works, and you want to generate some basic knowledge about that system. So you need tools that allow you to take that system apart in order to re-engineer it for a particular purpose or phenotype that you're looking for. So in order to fulfill the promises of synthetic biology, we really need high throughput tools to make diverse edits genome-wide. So if you look at a lot of the current tools that are available in the genome editing space, there's a lot of challenges. So very often they lack precision. There are slow and laborious workflows. There's a lack of diverse edit types. Mostly loss of function mutations are being introduced. And it's difficult to separate hitchhiker mutations from true causal mutations. And this is a classic problem for something like classical mutagenesis. Another problem is when you look at all these different technologies that are out there, you either have to sacrifice the number of genomic locations that you can target or the variety of edit types that you can introduce at those genomic locations. And what you really want to do in order to do true genome engineering or genome discovery is get up here into this quadrant where you're able to introduce a lot of variety of edit types at many different genomic locations. So towards that end, I'd like to introduce you to Inscripta's Onyx platform, which allows for programmable and trackable variation to be introduced into a genome, and currently we support E. coli and yeast. Okay, so the platform is truly a platform. It has a lot of different components to it, and it supports all aspects of the design, generate, test, learn cycle, which is kind of fundamental to the scientific method. So it all starts with the Inscripta designer software, which allows you to go in and basically design the edits that you want to introduce into the genome. You don't have to know anything about guide RNAs or design scores. The software basically takes care of all of that. After you finish designing your library, which can have up to thousands of different edits, that initiates the manufacturing of the reagents and the consumables that are necessary in order to build that library. In a matter of weeks, you will receive those consumables, which then goes into the second component of the platform, which is the Onyx instrument itself. You put the consumables in along with the cells that you want to build this library in, and you press go. And in a matter of 2 to 4 days, depending on if you're doing E. coli or yeast, you will get this tube of engineered cells that comes out of it. So this tube of cells contains basically thousands of different strains that were designed within the platform, and each cell will contain at most one of the edits that was contained in your design library. But it's a population of all of those edits within one tube of cells. Then this will go into the test aspect, which is this is what the customer supplies, either if you're going to do some sort of screening workflow or a selection type of workflow. After you identify your hits from screening or have enriched for your population through a selection, you submit that for DNA sequencing. That DNA sequencing data then gets imported into another package called Resolver, and this is what allows you to link the genotype of the edit with the phenotype that you've obtained. From this, you'll generate some information that will allow you to go through this cycle once again. So not only does the platform allow you to go through each aspect of the cycle, but I hope to convince you with the applications I'm going to talk about later that you can go through this quite rapidly. So the platform has optimized designs. It utilizes the proprietary MAD7 nuclease, push-button simple workflows, anywhere from 100 to 10,000 designs per library. There's a single edit per cell. You can introduce diverse edit types, which I'll talk about in a moment, anywhere from 1 to 63 base pairs. Also, there is a DNA barcode that's attached to each edit or associated with each edit, so you can do rapid genotyping through just sequencing of the barcode. And it really democratizes genome editing, basically bringing genome editing to the masses. So there's a wide diversity of edit types that you can introduce, and this can target both the non-coding region, say regulatory regions, promoters, terminators of genes, as well as the coding region itself. So we like to very often introduce knockout or produce knockout libraries where we introduce a triple stop codon towards the 5' end of a gene, or you can even do saturation mutagenesis, mutating one residue to all their possible residues within a protein. So lots of different types of edits you can introduce. Okay, so some of the specs. So we currently support E. coli and S. cerevisiae. This includes Inscripta standard strains as well as related customer strains. Again, you can target the coding and non-coding regions in the native genome, or if you've introduced a heterologous sequence, as you'll see in the applications I'm going to talk about, you can also target that as well. Designs per library for E. coli, it's 100 to 10,000, or cerevisiae 100 to 6,000 edits per design. One edit per cell. You can do insertions, deletions, and substitutions, anywhere from 1 to 63 base pairs. The library composition that comes out of the instrument, it's typically greater than 90% of the designs within your design library are contained within that population, and the editing fraction is anywhere from 15 to 75%, depending on the complexity of the edits that you're trying to introduce. As far as time, reagent delivery is approximately 3 weeks, and the library build is anywhere from 2 to 4 days, depending upon whether it's E. coli or yeast. However, the hands-on time with the actual instrument is less than an hour. Okay, so that's an introduction to the platform. Now I want to tell you about the iterative genome engineering workflow that has been recently released for the platform. So this is what the workflow looks like. So you build your first library, you take it through your screening workflow here, and you identify your top hits. You take one of those hits, and you want to carry that into the second round. So in order to do that, you'll need to cure the plasmid or plasmids that are contained within that strain that was necessary in order to introduce the edits. For E. coli, which has 2 plasmids, it requires an active curing process, and Inscripta provides a kit, an on-instrument kit that allows you to do that curing process. For S. cerevisiae, it's a passive process. So once you've cured your plasmid, that can go immediately into round 2, where you build another library on top of that strain, basically adding a second round of edits, going through your phenotyping workflow, identify your top hits, and then go through the process again of curing the plasmids and building your library. So it's a very simple workflow in order to do this. Okay, so now I want to tell you about some applications where we use this iterative genome engineering for a particular purpose. So this first one is where we wanted to do heterologous protein engineering. So basically do some protein engineering on a protein that we've introduced into the genome of E. coli in this example. So in this example, we used a classic GFP protein, which is a nice model system to use. We integrated it into the host genome with a strong promoter. This is an iGEM J23102 promoter, a strong ribosome binding site, and a T0 terminator. So once you've introduced your heterologous protein, there's lots of different ways you can design libraries to introduce diversity into that protein. So you can do SNPs, you can do insertions, you can do deletions, you can target the regulatory region or use regulatory data. You can use evolutionary and strain variants, any published enzyme or protein variants, or you can do structural guided variants. So in this particular case, what was done was utilize a database of fluorescence variants of GFP. So basically the superfolder GFP sequence was aligned against the published database, and hotspots were identified within the coding sequence. And basically a hotspot was defined as any place that had greater than one mutation at a particular location. So once those hotspots were identified, a saturation mutagenesis library was built targeting those hotspots, and in total there were 723 edits within that library. And basically those edits were agnostic to the mutation effect from the database. So after that library was built in E. coli, and you can see here's an example of some of the fluorescent colonies that were obtained after the library building. In total, 192 colonies were screened through this workflow. They were tested at 3 different excitation and emission pairs using either YFP, GFP, or BFP excitation and emission wavelengths. So here's a look at some of the data in the GFP versus YFP signal. So you can see there's lots of them that still have the wild type GFP signal. There are many of them that were disrupted for fluorescence, so those were not tolerated mutations, so that's not surprising. There was one hit that seemed to have a significant improvements in fluorescence signal in the GFP axis, and then there's also a YFP variant that was also identified. If you look at 3 axes now, GFP, BFP, and YFP, this is that improved intensity one that I mentioned. We also saw lots of BFP variants or BFP shifted variants. And one of those variants was a previously identified and well characterized a tyrosine 66 to histidine variant. So you can identify variants that was already known in the literature. This improved intensity variant was a phenylalanine 145 to methionine variant, and that was carried through a second round of iterative editing. So basically this variant then had that same initial library built on top of it, and then 96 additional samples were screened. And what was found here was that there is 2 isolates showed a high yellow shift. One of them had a very high intensity here, and this happened to be a novel YFP variant that hadn't previously been characterized. So this is just an example of using GFP as sort of a model protein. Obviously, you can substitute your favorite protein or any enzyme in its place, but just to show you an example that you can do iterative rounds of genome engineering within the Onyx platform in order to identify improved variants. Okay, now I want to tell you about another application where we wanted to improve the heterologous expression of a protein in yeast using this rapid iterative genome engineering. So protein production is important for the bioeconomy, and it's important for, say, industrial biotechnology where you want to produce biofuels or use enzymes in textiles or food production or processing. Obviously in biopharma or therapeutics, you can target metabolic disorders like diabetes, which is a big one with insulin, or in cancer therapies. And a really hot area right now is this alternative protein market where you want a non-animal source of, say, meat protein or dairy proteins or egg proteins. And really, the current tools to improve protein production are quite limited. So you can, one common technique is to insert extra copies of a gene within the genome. So this can definitely improve expression, but those improvements tend to plateau. You can tinker around with the expression cassette. So you can target the promoter or use alternate promoters. different terminators. You can change the gene design, codon usage or codon optimization. Or if your protein is secreted, you can try different signal sequences. And a very common way that's currently used is doing classical mutagenesis of a production strain. So this allows you to basically take your production strain, treat it with some sort of mutagen. This introduces tens to hundreds of different SNPs throughout the genome. It can introduce beneficial mutation as well as deleterious mutations simultaneously, so that's a problem. And it can be complicated to identify causal mutations for the improved production or the improved phenotype you're looking for. So what we think is that the Onyx platform provides a really attractive alternative to improving protein production. So we wanted to come up with an application to test this out, and we decided to use our model enzyme is an enzyme called CBH1. So this is a cellobiohydrolase 1. It's an enzyme that's involved in cellulose degradation. It has a lot of importance in this consolidated bioprocessing model, which basically you would take a strain that produces various enzymes. Those enzymes would degrade a feedstock, which is very often waste plant material. When that waste plant material is degraded, it produces sugar. Those sugar are then consumed by that same strain in order to produce some sort of fuel or some sort of chemical that's used for some industrial process. So this is the model we wanted to use. So we utilized a fungal CBH1 that was codon optimized for expression in yeast. And we used the native fungal signal sequence, so this protein would be secreted. We used the native yeast ENO2 promoter to drive expression of the CBH1 and a DIT1 terminator. So the base strain that was used was a strain called SenPK, and we generated a single copy genomic integration of this expression cassette. So the assay is pretty straightforward. We used this synthetic substrate called PNP beta-lactoperonoside. When CBH1 acts on it, it releases paranitrophenol, which has a characteristic absorbance at 405 nanometers. So we could very quickly measure the amount of active protein that is being produced by measuring the absorbance at 405 nanometers. And this base parent strain that was used for expression had no detectable activity on this substrate. So to show you a little bit about the noise within the assay, all of the screening workflow was done in a high-throughput format where we used 96-well plates, anywhere from about 10 to 20 plates of samples per library. So if I pick 96 colonies of this base expression strain, take it through the workflow, you could see the average and standard deviation of the measurements and a very low CV, which gave us pretty good confidence that we could pull out hits even if they had a very low improvement in CBH1 expression. Okay, so we wanted to test a bunch of different library concepts for trying to improve the production of CBH1. So this is the first on library strategy, and it really focused on a lot of different diversity. So we had genome-wide libraries, so basically targeting all over the genome. This included a genome-wide knockout library, so knocking out every gene in yeast, a genome-wide synthetic terminator insertion library. So we utilized a synthetic terminator and inserted it at the 3' end of every gene in yeast, and this can have an impact on the ultimate expression of that gene. And then we had another library where we basically introduced 1 to 59 base pair deletions kind of throughout the genome. And these deletions can target either the open reading frame itself of a gene or the regulatory regions of that gene. So those are our sort of genome-wide, unbiased, untargeted libraries. As well as the targeted libraries or rational libraries, we had 2 libraries that were targeting the ENO2 promoter that was driving expression of CBH1. So we had one library concept where we took the predicted transcription factor binding sites and either inserted extra copies of them or deleted them. Another strategy where we took those same binding sites and we swapped in various other yeast transcription factor binding sites at those locations. We also had a CBH1 alternate codon library. So this is basically a site saturation synonymous mutation library where you take at each position, you mutate those codons to all other possible codons that encode for the same amino acids. We're not making mutations, or we're not changing the amino acid. We're just changing the codon. I also had another library that I'm calling the targeted knockout library. And in this library, we basically mined the literature looking for genes that had been previously shown that when you knock them out, they improve the production of a heterologous protein. I also did a genome-wide protease deletion library, so host proteases can often be problematic for heterologously expressed protein. And then I also targeted genes in the glycosylation pathway for deletion as well, because they can have an impact on secretion of proteins. So these are the concepts that we tested. Okay, so to give you a feel for the size of these libraries and the number of isolates screened, there were 7 total libraries that were built, about 15,000 different edits designed within those libraries. And I screened about 8,500 isolates. I'm also showing the estimated library coverage from these different concepts. So this is really based on lots of different factors. One of them is the size of the library, the number of isolates that are screened, obviously, the editing fraction within that library, because not all cells are going to be edited, and also the distribution of edits within that population, because you're not going to have an equal distribution of all the edits within that population. So that's what's used to estimate the amount of library coverage that was screening these number of isolates. So you can see some of these libraries, some of the larger ones, are screened at fairly shallow diversity, 4% to 18%, whereas some of the smaller libraries, like the targeted knockout library, you're screening a fair amount, 2/3 of the diversity that's contained within that library. I'm showing the number of retest hits. So all of these libraries go through a primary screen. We take those hits we identify, they go through a retest in quadruplicate so that we can confirm that they're indeed hits, and then also get some statistics on those hits. And this is showing the number of unique edits from each one of these different libraries. So some of the smaller libraries where we do fairly deep screening, you can see that we're pulling out the same hits multiple times, or more often we'll pull out the same hits multiple times, whereas that's not true for some of the larger libraries. And in total, from all of this screening, we identified 74 unique hits from this primary screen. And I think one of the important points I want to bring up here is that even though we have these very large libraries and we're only screening a fraction of the diversity that's contained within those, we're still able to identify a significant number of unique hits. And ultimately, this is the important part of your first round screening, is diversity generation, to identify diversity that you can then combine in subsequent rounds of genome engineering. And that's what we wanted to do. So shallow screening of large libraries can be very effective at identifying diversity that you can then combine in subsequent rounds. Okay, so here's what the data looks like. This is all retest data for all the hits that we identified. And I'm showing the full change over the base strain. So the full change in CBH1 activity over the base strain. And I colored it by these different library concepts, so all 7 different library concepts. So you can see that with the genome-wide and the untargeted, basically untargeted or unbiased approach, we were able to identify a lot of hits. And then from the targeted libraries as well, we were able to identify some nice hits, anywhere from about a 1.1 to a 1.6-fold increase in CBH1 activity. This is showing the average of the quadruplicates as well as the standard deviation. If we pull out a hit one time from the library, if we pull it out multiple times, then we average those hits and got the standard deviation from those hits. And you can see that there's fairly low noise within the assay, which gives us pretty good confidence in the fold improvement that we're obtaining. Okay, so this is all the primary screening, or the first round of screening, I should say. So now, we identified all this diversity. Now we want to start to build on that diversity. And we decided to use 2 different strategies in doing that. So for the second round, we wanted to do a targeted type of library as well as an untargeted type of library. So again, that's what we did in our first round, right? It's an unbiased approach, an untargeted approach, and then a targeted type of approach, or a rational type of approach. So what we did is we took 2 first round hits. I wanted to see if there was any difference, and depending on which hits we picked, and we built a genome-wide knockout library, the same genome-wide knockout we used in our first round. After that, we identified a top second round hit, and then we built another library on top of that. And in this case, we used all the first round hits we identified, and we stacked it on top of that second round hit. So that was one path. And we identified our third round hits. In another path, we took a first round hit, the same one that we used up here, and we took the first round hits and stacked them on top of that. So it's basically the hits on top of a hit. So those are the two strategies we used. Okay, so here I'm showing the primary screening data from the first round libraries, the second round libraries, and the third round libraries. And this is showing the fold improvement in CBH1 expression over the base strain. So the first round hits, again, all those different library concepts that we identified. We took actually 2 hits from this first round and carried it to the second round. And the first thing we did, as I mentioned, is we built a genome-wide knockout on top of them. And that's what's shown here. So those are these 2 things. And you can see that there's a slight difference in basically the base strain performance that we used, but we got nice improvement from these genome-wide knockouts stacked on top of that. And it was what we pretty much showed is that the fold improvement over the base strain, so like going from here to here, is kind of similar from here to here. So basically the fold improvement didn't really depend on which base strain that we were picking. And what we decided to do to carry through the third round was basically let's take our top hit into the third round. In this library here, we took this same hit from the first round and we stacked all the first round hits on top of that library. And what you can see is we see, again, a nice improvement in CBH1 performance. So stacking libraries on top of hits from the first round caused an even greater improvement in CBH1 expression. So we took this first round hit, and then we took all the first round hits and stacked it, or sorry, this is a second round hit, and we stacked all the first round hits on top of that, and that's what's shown here, again showing that we see a nice improvement in overall CBH1 production. So through iterative rounds of genome engineering, we're able to gradually increase and improve the expression of CBH1. Okay, so now this is all the retest data from the first round, which I've already shown you, the second round, and now the third round to kind of show you what does the retest data look like. So these 3 different libraries from the second round, you can see that there's a nice improvement in CBH1 expression, and then we saw a significant improvement into the third round. Again, showing the standard deviation of quadruplicate measurements, or if we pull out the same hit multiple times, we average those. So you can see there's very low noise within this screening workflow in the high throughput, the screening strategy that we used. So overall, these different rounds, we were able to continue improving CBH1 expression. Okay, so we built a lot of libraries that were basically targeting all over the genome, whether it be a genome-wide knockout, deletions across the genome-wide terminator library, or a targeted knockout library. So all of those libraries were basically targeting lots of different places in the genome. And we pulled out a lot of hits doing this strategy. And what I wanted to see was there any commonality between the genes that were targeted for these different library strategies? And then when I kind of categorized them by different cellular processes, I saw something interesting. So if we look at protein degradation, we identified a lot of hits that target genes in protein degradation. Hits targeting protein trafficking, cellular stress response, transcription and translation processes, nuclear transport, glycosylation pathway. Some of them didn't really fit into any of these. I kind of lumped them all together into other. And also interestingly, we identified 11 hits from these strategies were genes of basically unknown function. This was pretty exciting to us because using this sort of unbiased or untargeted strategy, allowed us to pull out genes of unknown function, which you likely would not target through some sort of rational library building strategy. So we identified all these hits in all these different cellular processes. And I think what was kind of exciting to us, looking at these is, hey, if I think about heterologous protein production, these are all the pathways that I think might be important for that. You don't want to degrade your protein that you're expressing. You need to make sure that it's being trafficked, in the proper way through the secretion. This is probably stressful to the cell in order to overproduce a protein. You need to optimize transcription and translation machinery, nuclear transport. So it was exciting to us that these are all processes that you might imagine to be important for heterologous protein production. And in total, 69 unique genes were targeted for edits from these hits that we identified from these various different library strategies. Okay, so that is all the high throughput screening data. And we said, okay, that's great. If you look at what happens in a 96-well plate with 300 microliters of media, you can identify these hits. Well, what happens when you scale it up? So what we want to do is at least scale it up to shake flask cultures. So we decided to scale it up several of the hits to 25 mil shake flask cultures using the same media, YPD, with glucose that was used for the high throughput screening. And we identified, or we picked, three different hit strains from each round. So basically 9 different hit strains, and then compared it to our base strain. And that's what's shown here. So first I'm showing just the growth curves. So this is basically just looking at the absorbance at 600 nanometers over time. So taking an early time point, about 6 hours, 24 hours, 48 hours, and 72 hours. Again, 3 strains from the first round, 3 from the second round, 3 from the third round, and then the base strain for comparison at this is shown as this dashed black line. And I think what was interesting and immediately popped out at us is, hey, all the hit strains we identified, they basically have decreased growth relative to this base strain that we were using. So this was interesting in 2 ways. One of them is, excuse me, that we're seeing improved production of CBH1, and since we're not seeing greater growth than the base strain, it means that this can't be explained by just better growth of those strains. In fact, they grow slightly worse than the base strain. And another interesting point is that there's been reports in the literature that very often strains that have improved heterologous protein production have decreased growth relative to, say, a base strain of expression. And this kind of makes sense if you think about it, because more amounts or greater amounts of the carbon is being diverted towards protein production instead of towards biomass production. So our results, kind of fit with what's been reported in the literature, and it's interesting that all of our hits from the different rounds basically grew worse than the base strain. Okay, so that's growth. Now what we really care about is, well, what about CBH1 expression and CBH1 production? So that's what's shown here. Again, the base strain is shown here as a black dotted line, and it was very nice for us to see that our first round hits clustered near each other with improved production over this base strain. The second round hits also showed improved production over the first round hits, and they clustered together nicely. And then finally, the third round hits had the highest level of production, and they clustered near each other. So there's this one variant here, which is one of the third round hits, which actually, if we go back, had kind of delayed growth here. Looks like you also have delayed CBH1 expression up to about 24 hours. Then when you get to 48 hours, it pops right up and has much greater CBH1 expression relative to these other strains that were tested. Okay, and then the top hit that we identified basically had a 4-fold improvement in CBH1 production over the base strain, pretty much at the, I think, 24, 48, and 72-hour time points. It was about a 4-fold improvement. So significantly improved CBH1 production over these 3 rounds of genome engineering work that we did. Okay, so this is showing you the data, right? So this is the types of improvements that we were able to get through these 3 rounds. Now I want to talk to you about, well, what's the time investment involved in doing this, and the work investment, the FTE requirement in order to do this type of work. So I wanted to go through this basically project timeline. So I broke it down into different steps. So first you have to design the libraries and manufacture the reagents. Then you have to build the libraries on the instrument. You have to phenotype and genotype those libraries and the hits. So this takes about a month time. Then if you're doing iterative rounds of genome engineering, you'll have to cure the plasmid from the hit that you carry to the next round. And then you can either design a new library, like we did with the CBH1 hits on hit library. So we had to take all those hits, design a new library, and manufacture reagents to build that library. Or in the other strategy, we used a genome-wide knockout library. So that library we had designed, we had multiple kits of that particular library. So we were able to immediately go into the second round if we use that library that we already have. And this is a strategy that you can use designing new libraries as well as recycling libraries that you've used in the past. And then those will then obviously go into your screening workflow. Okay, so here's the project timeline. So the first round of screening work that we did, that took about 3 months. So it was about 3 weeks to design and manufacture the reagents. It's about 3 to 4 days to build the libraries because this is using yeast. And then it's about a month time to phenotype and then genotype those 7 different libraries that were used in the first round. And we could do, the library builds had to be done in series because you only had one instrument, but you could do the phenotyping in parallel. Okay, so in total, to identify those 74 unique hits from the first round, it took about 3 months. Okay, after doing that, we picked a first round hit. You can take one of your first round hits, and then you have to cure the plasmid. So the curing procedure takes about a week in order to cure the plasmid and then QC that strain in order to make sure that indeed that plasmid has been cured. Because if the plasmid is not cured, your library build is not going to work very well. So you cure that plasmid from that hit strain, takes about a week. We decided to use a genome-wide knockout library first, since we had that on hand, so that could immediately go into your library build, which takes, again, about 3 to 4 days. And then you could go into the screening and phenotyping and then genotyping those hits. So the second round took about 1.5 months. At the same time, we were manufacturing, we designed and manufactured the first round hits. Library. So we took all the 74 hits here, threw them into one library, and we built that first round hits library, or manufactured the reagents for it. So then we went into the third round. So we took one of our second round hits, we cured the plasmid from that strain, again, that takes about a week, built the library, 3 to 4 days, and then this is a first round hits library, and then we phenotyped and genotyped that. So the third round took about 1.5 months as well. So in the span of about 6 months' time, we're able to identify a top strain that had 4-fold improvement in CBH1 production. So a significant improvement. Now, obviously, this timeline can change a little bit depending on your strategy that you used. So 7 libraries in a first round is a fairly large number of libraries. So you could probably cut that down to a smaller number and decrease the amount of time that's spent in this first round, which would shorten this total time point, this total timeline for the project. Okay, so that's talking about time. Well, what about effort? So FTE time. So that's actually quite minimal. So designing these libraries takes anywhere from a half an hour for very simple libraries to a few hours for some of the more complicated libraries. So not a huge amount of investment in order to design these libraries. So building the libraries or curing the libraries, this is really minimal effort. It's really a few hours per week in order to put the reagents on the instrument, press go, or go through the curing procedure. And then phenotyping or genotyping is about a quarter FTE in order to do that work. So a lot of the effort is actually spent phenotyping your libraries. So while you're able to go through these iterative rounds of genome engineering very rapidly, it also doesn't require a significant amount of resources time resources or people resources in order to do that. Okay, so as far as conclusions, so the Onyx platform is really a game changer for DGTL cycles. So it allows you to rapidly design and build genome-edited libraries using significantly less resources. And it truly democratizes genome editing. So I like to use myself as an example when I talk about this, because prior to coming to Inscripta, I had never designed a library. I had never built a library, but I had phenotyped a library. But now with the Onyx platform being available, I'm able to come up with an idea or a concept that I want to test. I can go into Designer software very easily and rapidly design a library testing that concept. Once the reagents come in, I can put those into the instrument along with the cells and press go, and then build that library. And once that tube of edited cells comes out, I can immediately go into phenotyping. So I can do all of that on my own. I don't have to, I have to have very little skills as far as bioinformatics or molecular biology skills. So it really kind of revolutionized the type of work or how I do my work. Another important point is that iterative genome engineering with the Onyx platform allows for rapid development of proteins, improved proteins. So, the GFP example, as well as strains with significantly improved phenotype. So, the CBH1 expression phenotype. And that alterations, this is for the heterologous protein, is that alterations in diverse cellular processes can impact heterologous protein production. It was kind of amazing to me that we identified 69 different hits throughout the genome that could improve CBH1 production. So it really suggests that there's lots of different pathways and lots of different genes that are involved in heterologous protein production, lots of opportunities to optimize your strain and improve the production of your protein. And with that, I'd really like to thank you for your time in being able to talk to you about the platform and some of the applications that we've done using the platform, and I'd be happy to answer any questions that you might have.
Thank you, Eric, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, can you estimate the metabolic burden in heterolog production as a factor or any other metric?
Yes, very good question. So that's not actually something that we looked into in any level of detail. I mean, based on the growth curves, it appears that there definitely is some sort of burden that's putting on the cells when you produce a heterologous protein, based on the fact that the cells seem to grow more slowly or to less of a degree overall. So there is some level of metabolic burden, but we didn't do any type of analysis to try to estimate what that burden is or to look into that more fully at all. We had some ideas about trying to direct some of the flux of the carbon towards, say, amino acid production, but we didn't really test out any of those types of concepts within this project that they were doing here. But very good question.
Thank you. Next question. You said that the CBH1 gene was optimized for expression in yeast. Why do you think you were still able to identify hits in the alternate codon library?
Yeah, so that was actually a concept that I wanted to test. There's some reports in the literature that if you introduce say, rare codons at certain locations within the gene, you can actually improve the expression of a heterologous protein. And the idea is that when you're producing a protein, you may not want the fastest translation rate possible because very often there are places within the translation time where you may want the ribosome to pause or to slow down to allow for a subdomain to fold properly. So what was found in the literature is that you can sort of over-optimize or over-codon optimize a gene so that you get very rapid expression of that protein, but you may not get the most amount of active protein in the end, and that some of them might be aggregated or insoluble or basically not folded properly. So it came out of that concept, but instead of just making, say, rare codon introductions throughout the gene, I said, hey, this is very easy enough to do using the Onyx platform to really design a library where you can just do a site saturation synonymous mutation library. So I went in and used Designer to very rapidly design that type of library. So the reason I think that you can pull out some of these hits, even though the gene is codon optimized, is one, because of the translation rate that could impact the overall amount of active protein you get at the end. Another thing is that we may not know all the rules of codon optimization, and it could be that there are things that we don't understand as far as, say, mRNA secondary structure that could impact translation rates. And also, I can imagine that the pool of tRNAs might change during growth conditions. So maybe at the beginning of growth, you might have a different pool versus mid-log phase versus the end of growth, and that what you think you might be optimizing for overall as far as protein production, it might vary at different stages of growth. So those are, I think, some of the ideas that might explain why we're able to pull out hits in that codon alternate codon library for CBH1.
Thank you. Next question. How flexible or robust the designer software is when it comes to non-canonical, non-model microbial species? What are the inputs and outputs of the software?
Yeah, so currently the platform is optimized and really supports only using E. coli and S-servCA. So the software and the design scores and the guide RNA designs, that's all been optimized for use in E. coli and S-servCA, a couple different strains of each one of those. So it doesn't really support the use of other microbes at this time. This is something that we're really mindful of. We get a lot of input from people of, hey, are you going to support my species, my strain of yeast, or my strain of bacteria? So this is input that we're taking at Inscripta, and there are things on the roadmap as far as future species expansion. But right now, the platform really only supports the use of E. coli and S-servCA, and then the designer software, it also only supports E. coli and S-servCA.
Thank you. Next question. Can you further describe the use of barcodes? Are they incorporated into the genome?
Yeah, good question. So they're not incorporated into the genome. So they're actually maintained on the plasmid. So there's a plasmid that is used for editing in yeast. There's multiple plasmids for E. coli. So they're actually not incorporated into the genome. They're maintained on the plasmid. So if you want to do sort of like a selection type of experiment where you grow, say, a population, maybe there are tube of edited cells that come out of the instrument under some sort of selective pressure, you can do that. And then at the end of your selection experiment, you can just sequence the population of cells that are contained in that, where some edits might have caused an enrichment in certain strains, whereas other edits might have caused a depletion. And you can basically just sequence the barcode. And basically the barcode is a proxy for the edit that's contained within that cell, and the number of that barcode is sort of a proxy of the number of cells within that population that contain that particular edit. So yes, so to answer your question, it's not integrated into the genome. It's maintained on the plasmid.
Thank you. Next question. If you have multiple copies of the same expression cassette in the genome, will the edits designed within the cassette actually been done to all these copies?
Yes, another good question. Because yeah, for heterologous protein production, very often you have multiple copies of a gene. So to answer your question is you will get editing at some number of the locations. So biology says that you put a guide in there and you put a repair template, you're going to get cutting or potentially cutting at multiple sites, and you can get editing at multiple sites. But it's not clear, you won't necessarily get editing at all of them, or you might get editing at none of them, depending on how good your guide is and how good the editing actually takes place for the repair. So I will say that we've tested this out at multi-copy or basically multiple locations where there's multiple copies of the same gene. And sometimes we get editing at both locations. Sometimes we get editing at only one location. Sometimes you're getting at none of the locations. So you can get quite a mixture of cells at the end. I think for the most part, we saw that we got editing at multiple locations when we tried this type of experiment. Now that's with two loci. So you can imagine as you increase the number of locations, 4, 5, 6, 7, and 8, and if you're actually introducing cuts at all of those different sites simultaneously, that can be very toxic to the cells. And it's probably going to dramatically decrease your editing efficiency and frequency if you try to introduce cuts at many locations. But a handful of locations, maybe it's going to be okay. We definitely see that it becomes more toxic to the cells at 2 sites. 3 sites definitely is much more toxic. So you kind of have to keep in mind the number of locations that you're targeting. Currently, the software actually only supports editing at 1 location, but it's something that we're planning to enable soon that will allow editing at multiple locations. So basically multi-copy editing within the same genome.
Thank you. Next question. In the 3rd generation strains, what was the amount of protein in grams per liter in the shake flask tests?
Yeah, that's a good question. So I haven't actually gone to measure the amount of total protein that's being expressed. So it requires me to purify some of the protein and then have a standard that I can use sort of to quantify the amount. I haven't done that experiment yet, but it's something that I definitely want to do to kind of get an estimate of the amount of protein that's being produced. But I don't have an answer to that question right now.
Thank you. Next question. Do you think that with ALE, the 3rd round hits could be adapted to grow at the same rate as WT? Any thoughts around the cells adapting to the Onyx microenvironment during transformation and library screening?
Yeah, so I think that's kind of 2 questions. So one of them is, I guess, can we improve the growth of the cells, these 3rd round strains, using an ALE type of experiment? And I think that's definitely possible. That something like that could potentially work. I think the other question is related to what happens, on the instrument in the microenvironments when editing takes place. And yeah, so definitely there's a lot of biology that's happening on the instrument, and it's not clear exactly. There could be all kinds of things that are happening, and the cells are adapting, to those microenvironments. You're putting stress on them by inducing editing. You're inducing cutting and then editing. So there's definitely some stress that can be happening to those cells. There's not a clear picture of what exactly is happening on the instrument in those microenvironments, but definitely it'll be a somewhat of a stressful environment for the cells.
Thank you. Next question, and it could possibly be a 2-part question. So, do you confirm the loss of plasmids before phenotyping, and if not, do you see any heterogeneity in its phenotype?
So the first question is, do we confirm the loss of the plasmid? So in these types of experiments that we're actually doing, we actually maintain the plasmid. So I grow it under selection to maintain the plasmid. You can definitely do phenotyping so that you don't maintain the plasmid and that you lose the plasmid. The plasmid is not required in order to do your genotyping at the end. But in the experiments that I described here, I actually did grow those cells under selection to maintain the plasmid. And I was able to identify those hits. What happens when you remove the plasmid, I don't know at this point. Maybe you get greater expression of the protein, because definitely maintaining that plasmid can put some metabolic burden on the cells. And then what was the second part of the question?
The second part was, if not, do you see any heterogeneity in its phenotype?
So I think the question is, do we see heterogeneity within a sample with the phenotype? And that we don't. It seems like it's pretty stable as far as what we're seeing as far as the plasmid. But if the question is, if we lose the plasmid or remove the plasmid, what happens to protein expression? That I don't have an answer to at this point because I haven't tested that.
All right, thank you. Next question. Could this tool be applied to other yeast model organism as Pichia pastoris? Is it Pichia or Pichia?
Pichia pastoris.
Pichia, okay.
Yep. Yeah, good question. So as I mentioned, so we're currently supporting yeast, ester of ECA as far as yeast. We do hear a lot of interest in Pichia, so that's probably one of the most common organisms that we hear that people have interested in using on the Onyx platform. So we're definitely mindful of that and thinking about that as far as future expansion of the platform to new species. So Pichia is definitely one of the hot ones that we hear a lot of interest in. But at this point, it currently does not support Pichia.
Great, thank you. It looks like we have time for one more question. Do you have to use whole genome sequencing to find out which mutations are changed in the libraries?
So you don't have to do that. So there's actually a few different genotyping techniques you can use. So one of them is you could just, if you maintain the plasmid, you could just read the barcode. You could sequence the barcode, Sanger sequencing, and figure out which edit that correlates with. If you want to look at to make sure that the edit is actually introduced into the genome, you could do barcode sequencing to identify what's the edit that's supposed to be in that gene or in that strain. And then you could do Sanger sequencing of the genome. So basically do PCR amplification of the genome around that location, and then do Sanger sequencing to identify to confirm that the edit is actually in the cell at the location that you want. Or you could do whole genome sequencing. Whole genome sequencing tends to be the most expensive way to do it, but there's lots of different strategies you can use to do your genotyping and to confirm that the edit is actually introduced into the genome. But a good question.
Great. Well, thank you again, Eric, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Inscripta, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.